Hello, welcome. My name is Aabid Hussain, and today's topic, we are going to learn about one of the important topic related with the developmental biology, which become a part of CSIR-NET competitive exam. Okay, so we will see that this topic is related with the CSIR-NET exam, and comes under a developmental biology, right? What we are going to learn over here is about one of the reproductive structure called as vulva, okay? And the model organism that we are going to learn is a nematode called as C. elegans, okay? So let us further go in detail about this specific organism. Now, the point that we have to consider over here is at least the introductory part of the name of this specific organism. So basically, they are Asclepianthus or comes under the phylum nematodes. Okay. Now, point number 2 is that how they are maintained. Maintained means they can easily be grown over the culture of bacteria. Okay. So the point number 2 that we can include over here, they can be cultured by providing the food in the form of the E. coli colony, food source. Point number 3 is related with their sexes, right? So they are basically representing the hermaphrodite member. Hermaphrodite. Those who don't know about the hermaphrodite, let me tell you that they represent both as a male as well as a female. But early during the developmental stage, during the embryonic stage, they start to develop as a male. Later on, they develop ovaries. Due to the development of ovary, they gain the capacity to produce egg. So by this way, we can conclude that they become the hermaphrodite once they become the adult. Okay. Now, why it is so important? The idea of gene manipulation through RNA interference, we have studied a lot of information related with this C. elegans by applying the concept of RNA interference for genetic manipulation or gene manipulation. Okay. Now, this is the overall idea about the C. elegans or important, you can say the introductory part of C. elegans. Now, let us talk some important stuff related with vulva. Okay. Vulva is made up of, or it's made from 22 adult cells. Adult cells. These 22 adult cells will develop from 6 precursor cells, which we call vulva precursor cells. Okay. Now, the key concept which we are going to learn over here is related with the concept of induction. The concept of induction. Then we will learn the concept of lateral inhibition. Lateral inhibition. Okay. Now, one more important stuff is key players. So who are those key players? The key players are some ligands, that means some proteins as well as their receptors. So key player over here is point number 1, the signaling molecule called Lin3 molecule. The ligand of this Lin3 is Let23. Okay, Let23. Another key player is Lin12, and one more key player over here is Lin39. Okay, these are the major key players in this vulva development. All right, let us see about these one by one. But before that, we have to understand the body organization of this C. elegans. I will draw a rough sketch over here just to give you the basic understanding of how it resembles to. Let's say I have drawn this region as the anterior region of the body, and this part is the posterior part of the body. Location of the gonads. Gonad is located over here. And adjacent to the gonad, and you can say attached to the gonad, is the major important cell which we call anchor cell. And this anchor cell is very important, because it will become the source of induction. All right. Now, this will become the source of induction. Who is getting induced, right? So the one that is getting induced is the 6 precursor cells which are located hypodermally. 4, 5, 6. These 6 cells, they are called as, or they are named as, vulval precursor cells. All these 6 cells, they will make one equivalence group. What will they make? They will make one equivalence group. They are said like this, they are said to be as equivalence group because all these cells, individual, every single cells of this one, they are having the capacity to give rise to vulva. That means that is the reason why they are said to be as vulval precursor cell. You know, identity of being precursor cells or vulval precursor cell is defined by the concentration of a specific protein which we call Lin39 present in all these precursor cells. Okay, Lin39 in all these VP6 VPCs, which maintains that they are to be called as vulval precursor cells due to the presence of Lin39 protein inside all these cells. Now, another concept is that, yeah, about the names of these cells. These 6 cells, they are named so, like P3P, P4P, P5P, P6P, P7P, and P8P. These are named so. These are laterally arranged from left to right. So this is considered to be as the first one. If I'll just elaborate this structure, so the arrangement will be somewhere like this way, from P3P, P4P, P5P, P6P, P7P, and lastly P8P. Okay, they are arranged in this way only. That means from left to right. All right. Now let us consider the arrangement of these things in further detail. If I will draw this thing further, that means I have drawn the gonads in a zoomed version. This is the zoomed version of the gonad. Okay. So by the way, where is gonad located? Here's the gonad. Okay. What is the name of the cells which are associated with the gonad? We call it as anchor cells. So this is the anchor cell. Okay. And these I have already mentioned that they are the VPCs. Now, this is specific structure is the gonad, and associated with the gonad is the anchor cells. And as I have said earlier, just below the anchor cells, all the 6 VPCs will be present. Okay, so from centrally 2, 3, 4, 5, and 6. Now, they will be named accordingly. I will just give the numbers. Okay, so this is 3, 4, 5, 6, 7, and 8. Now the concept or the idea over here is that this anchor cell will release a specific type of protein which we call Lin3 protein. So here it is the Lin3 protein. This will be provided by anchor cells, and this will go and spread over in the nearby vicinity. Okay, nearby vicinity. You know, the fate is decided by the relative concentration of Lin3 protein that are being distributed among the different VPCs. You can see that cell number 6, which is P6P receives the maximum amount or maximum concentration of lin-3 protein, right? One more thing you have to understand is that if there is a ligand or the protein coming, then there must be someone to receive. There must be a receptor. So receptor to lin-3 protein is the LET23. So LET23 is acting as receptor. Alright. Now let us see how things are going on. As I have mentioned that number 6 cells or P6P will receive the highest amount or highest concentration of lin-3 protein. These are the lin-3 protein. Okay. So what will happen? The fate will be decided accordingly. How the fate is decided? Let us see over here. The cell number P6P, this is the cell P6P, and as it got the highest amount or highest concentration, this will develop into primary or 1 degree VPC. Okay, primary or 1 degree VPCs. Other cells which is neighboring, which have received comparatively less concentration of lin-3 protein, so they will develop into adjacent or lateral cells, or they will also be called as 2 degree cells or 2 degree VPCs. So this is 2 degree VPC. Okay. Now let me name this thing P5P and P7P. Out of the three, the central one can also be called as, this is also called as VPC, secondary VPC will also be called as lateral cells or lateral VPCs. Here also lateral VPCs also be called as secondary, and this one is also called as central VPC. Right? Now let us see, after receiving the signal in the form of lin-3 protein, what are the fate? The fate is that 1 degree of the primary cell will give rise to 8 adult cells. 8 adult cells. Whereas lateral cells will give rise to 7 adult cells. So similarly over here, 7 adult cells. So I mean to say that all the entire thing that means all the cells that will constitute 22 cells which will give rise to vulva. So simply vulva is made up of 22 cells. Now let us understand the signal transduction, or you can say the signaling pathway, that is very important with respect to the examination point of view. Whatever I have discussed so far, that is related with the concept, the basic idea. Now the signal system. First of all, signaling starts from lin-3. Lin-3 protein produced by anchor cells. This lin-3 protein will be received by the receptor present on this VPC. They are called LET23 receptors. Now once the LET23 receptor will receive the thing, okay, and this will give rise to that it is going to form primary VPC, or you can also call it as 1 degree VPC. Now what else is there? This primary VPC will release some substance or some compound which is going to activate the specific protein called lin-12, which is present in all the VPCs but in the inactive state. The signal coming from the VPCs will activate lin-12, okay, which will further inhibit them to become 1 degree, or which will inhibit them to become the primary cells, so that the fate is decided. So lin-12 will suppress the activity. That means it will not allow the formation of the primary VPCs, it will let them to become secondary. Okay, so the fate is also decided over here. So what is the idea that you have got from here? The idea that you have got from here is that it is the lateral inhibition and here induction. Clear? Secondary VPC. Now, what else is there? This lateral inhibition will cause the formation of the secondary VPCs over here also. So this is secondary VPC. Secondary VPCs will further restrict. What to restrict? Once they will become the secondary VPCs, that means they will not allow the expression of LET23 receptors. Okay, intracellular signaling. So within the secondary VPC, they will not allow the development of LET23 receptor, so that they will not further receive this Lin3 protein, right? As a result of that, their fate is decided. Okay, let us see some previous year question. So here what do you see? In C. elegans, an anchor cell and a few hypodermal cell take part in the formation of vulva. One important point I just forgot to explain you about the fate of these 3, 4 and 8 cell, right? 3, 4 will become hypodermal cell as usual, and here also hypodermal cell as usual, and these 3 will become primary and secondary vulval cells. Now let us come to the question. In C. elegans, the anchor cells and a few hypodermal cells take part in the formation of the vulva. That is true. The experiments performed to understand the role of these cells in the vulva formation and the results obtained are as follows. What is the result? If the anchor cell is killed by laser beam, hypodermal cells do not participate in the vulva formation and hence no vulva developed. That's true. Okay, because anchor cell is the source of induction. If the 6 hypodermal cells closely located, 6 hypodermal cells closely located to the anchor cells called vulvar precursor cell are killed, that means going to kill all the 6 VPCs, what will happen? The result is no vulva, right? Third is, if the 3 central cells, which 3 central cells? Come on. 3 central vulvar precursor cells are destroyed, the 3 outer, which normally form the hypodermis, take part in the fate of vulvar cells instead, right? So 3 central cells, what were those? The 3 central cells, they were the P6P and P5P and P7P, these 3 central cells. And then they are having some hypodermal cells like P3 and P4, and that side P8. Now, based on this information, following other certain statement regarding vulva formation, okay? Statements regarding vulva formation, anchor cells act as inducer. Do you find these statements correct? But what is the demand of the question? First of all, read the question. The question is over here. Which combination of the statements, above statements have been derived from the above experiment result? This is the important point, above experiment result. So this is the experiment result, right? And you have to figure out the statements from here that which are considered to be related with the experiment result. What are those? First is anchor cell act as inducer. No doubt, that is for sure. So here it is clearly mentioned, if the anchor cell is killed, that will not allow the formation of the vulva. So that is for sure true. So we will see out of the 4 options, the one which is having A will be considered. So by this way, 3 and 4 cannot be the option. And by the way, while practicing this question, always go through the elimination method first. So 2 options are being eliminated. Now you are having 50-50% chances. What is the difference between option 1 and option 2? Just the difference at the level of C and D. We'll see that. 6 hypodermal cells with the potentiality to form vulva form an equivalence group. What is the statement number 2 over here? See, if 6 hypodermal cells closely located anchor cells called vulvar precursors are killed, no vulva develop. That means if they will not be killed, they can have or they will have the tendency to develop as vulva. So statement number B is also correct. B is also correct. So still we are having the relative with these two. 3 out of 6 hypodermal cells participate in the vulva formation. I have said earlier that all the 3 cells which will give rise to 22 adult cells, who are they? P5P, P6P and P7P, right? This is what one is called as one is said to be as the primary and the 2 will be called as the secondary. What is mentioned over here? Statement number 3. You can see from here only. If the 3 central cell vulvar precursors are destroyed, the 3 outer cells which normally form the hypodermis takes the vulval cell instead. It simply means C is also related with the statement number 3. Okay. Now for our understanding, let us consider D. The central cell functions as a 1 degree cell and the 2 cells on the lateral side act as secondary cell. That is true, but the information is not provided in the experiment, right? So we will only consider option statement number A, B and C. Hence option number A. Now second question. Development of vulval cell in C. elegans is initiated by the induction of a small number of the cells by short range signaling from a single inducing cell. Hence that is called as anchor cells. With reference to the following statement that put forward. When the anchor cell was ablated means changed or destroyed and so on, early in the development no vulva is formed. Statement is true, but we will find which of the above statements is true. That means more than one statement is considered to be true. Okay, so this is true for sure. We will see that accordingly option 4 cannot be considered because they don't have statement number A. In the dominant negative mutant let23, a primary vulva, mutant of let23, a primary vulva formed, but the secondary vulva formation did not take place. Do you think that? Primary vulva formation? What is that? Let23 receptor. So this is the receptor. If the receptor will not be there, how come they are going to receive the signal in 23? So statement is wrong. That means B is wrong. So by this way only you can easily identify that statement A and B combination, this one is wrong. Although we are still having option number C, statement number C and D. Let us see. A cell adopting a primary fate inhibits the adjacent cell from adopting the same fate by lateral inhibition involving Lin39. Lin39 is for specificity of being equivalence, not by inhibiting. By the way, which was that protein? Yes, Lin12 protein. It was Lin12. So by this way we can say statement C is also wrong. So A and C is also wrong, so hence not this option. Now we are the last option, we are having A and D. Let us see even D options. A constitutive signal from the hypodermis inhibit the development of both primary and secondary fate, but it is overruled by the initial signal from the anchor cell. I have told you earlier as well that this Lin39 will be acting upon all the 6 vulval precursor cell VPCs that will restrict them further to become vulva. But once the signal is coming from anchor cell, it will be overruled. Alright. So hence statement number, the option 3 is considered to be correct one. The last question. During vulva development in C. elegans, the anchor cells produce Lin3 protein which interact with the Let23 protein present on the 6 vulvar precursor cell. That's true. That form an equivalence group. Okay, group. The central lineage P6P adopt the primary fate. Okay, good statement. The adjacent VPCs P5P and P7P adopt the secondary fate and the rest VPCs adopt the tertiary fate. That means going to give rise to hypodermal cells. Okay, fine. Few mutants in column A. In column A you are having the mutants and phenotype the result in column B. Okay, listed in the table. You have to match the column. That is very simple. Loss of function of lin-3 protein. Loss of function. What do you think? All VPCs adopt tertiary fate. Obviously, when there will be lin-23 protein will lose the function. So by default, they are having the tendency that will develop into hypodermal cells, and the production of hypodermal cells is as a result of 3 degree VPCs. Okay, so A with 4. A with 4, we are having these two options. Let's remove these two. Now, loss of function of lin-3 and gain of function of let-23. You know, two different types of mutation. Loss of function and gain of function mutation varieties. Okay. Loss of function let-3 and let-23. We will see further. Reduced function of lin-23. The function is reduced. Function is reduced. That simply means the one which is very near to the anchor cell will receive, at least that will receive the lin-3 protein. Okay, reduced function. It is not the complete loss of function. Reduced function. So the result will be P5P, P6P and P7P adapt the primary fate. P4P and P8 adapt the secondary fate. Is this the condition? Is this the condition? No, because you know this will only happen once the lin-3 protein will be distributed excessively among all the 6 members. So by this way you can say overexpression of lin-3 is associated with even P5P, P6P, P7P will adapt the 1 degree VPCs, and P4 and the P8, the most terminal, the most lateral side, they will adapt the secondary fate. So D is considered to be related with 1. So D1 over here. Okay. Again, for our understanding, let us see. Loss of function of lin-3 and gain of function of let-23. What will happen? Multiple vulva. I have said earlier that let-23 is maximally present on all the, that means the more and more receptor will be there. Okay, when the more and more receptor will be there, they have the tendency that a slight release of the you can say that inductive protein will further lead the formation of the multiple vulva. Okay, so by this way the combination statement A is correct. I hope that you have enjoyed this session. So please feel free to subscribe to the channel, and if you have any queries you can just mention your comments below. Okay, and don't forget to subscribe and press the bell button. Thank you.